Motor neuron, on reaching the target muscle, loses its myelin sheath to form a complex of 100 to 200 branched nerve endings, which are called nerve terminals or terminal buttons. Synaptic cleft, which is also called as junctional cleft, where synaptic neurotransmitter such as acetylcholine is released before it interacts with nicotinic acetylcholine receptors on the motor end plate. It contains acetylcholinesterase enzyme, which is responsible for the catabolism of released acetylcholine so that its effect on postsynaptic receptor is not prolonged. And third one is motor end plate, which is a thickened portion of muscle plasma membrane, which is sarcolemma, is folded to form depressions called junctional folds. These folds have nicotinic acetylcholine receptors concentrated at the top. Binding of acetylcholine to these receptors opens the channel, which allows the influx of sodium ions from the extracellular fluid into the muscle membrane. This creates a end plate potential and generates and transmits action potential to the muscle membrane. And steps involved at neuromuscular junction is uh, first one, an action potential travels down the axon to the axon terminal, results in electrical gated calcium channels opening, and this causes the calcium ions to enter into the axon terminal. Calcium causes the vesicles to release acetylcholine into the synaptic cleft, and acetylcholine diffuses across the synaptic cleft and binds to its receptor on the muscle. And on binding of acetylcholine, it opens ion channels and sodium ions rushes into the muscle. If the muscle reaches a threshold level on motor end plate, an action potential occurs on the muscle. And followed by the remaining acetylcholine will be broken down by acetylcholine esterase in the synaptic cleft. Neuromuscular monitoring, which is useful for maintaining adequate neuromuscular blockade and for the diagnosis of residual paralysis. And the conditions which we are using neuromuscular monitoring are after prolonged infusions of neuromuscular blocking drugs or when long-acting drugs are used, when surgery or anesthesia is prolonged, when inadequate reversal may cause devastating effects, and in renal or liver dysfunction where pharmacokinetics of muscle relaxants may be altered, and in neuromuscular disorders such as Lambert-Eaton syndrome or myasthenia gravis. The ideal nerve stimulator should be capable of delivering a variety of patterns of stimulations, including single twitch at 1 Hz, train of 4 twitch stimulation, which is usually 2 Hz with at least 10 seconds interval between the trains, and tetanic stimulation at 50 Hz for up to 5 seconds, and the last one is double burst stimulation. Good electrical contact with the skin can be established using ECG electrodes of silver or silver chloride variety. The ideal stimulator would also enable monitoring of the evoked responses. The most commonly used sites for nerve stimulation are ulnar nerve, where in that which we will stimulate and look responses in the adductor pollicis muscle, and second one is the facial nerve, in that we will look for we will use corrugator supercilii muscle, and in third one median nerve, and others are common peroneal nerve and the posterior tibial nerve. Single twitch stimulation. First one is the single twitch stimulation. In this single square wave supramaximal stimuli at frequencies ranging from 1 Hz to 0.1 Hz for a period of 0.2 milliseconds at regular interval, and the evoked response is observed. Twitch response will only be depressed when neuromuscular blocking drugs occupy 75% of the postsynaptic nicotinic receptors. And the most useful time to apply this stimulation is at the time of onset of neuromuscular blockade. Second one is train of 4 stimulation, which consists of 4 supramaximal stimuli at frequencies of 2 Hz given every 0.5 seconds. On using non-depolarizing muscle relaxant, there is reduction in the amplitude of evoked response with the T4 affected first, then twitch 3, twitch 2, and twitch 1. This decrement in twitch height is known as fade. During onset of non-depolarizing blockade, twitch 4 disappears at 70% depression of T, and twitch 3 disappears at 80 to 85% depression of T1, and twitch 2 disappears at 90% depression of T1. Twitch suppression of 90% would equal to a train of 4 count of 1 or less. Reversal of residual neuromuscular block can safely be achieved when train of 4 count is 3 or greater. Dividing the amplitude of fourth response by the first response provide the train of 4 ratio, it should be of 0.9 to 1.0 before tracheal extubation. And trainer 4 of 0.15 to 0.25 indicates adequate surgical relaxation. And in phase 1 depolarizing muscle relaxation, it will cause equal decrease in size of twitches but no fade. But in phase 2 depolarizing muscle relaxation, it will cause a fade of twitch. Tetanic stimulation consists of high frequency delivery of electrical stimuli. Example 30, 50 or 100 Hz. Most common is 50 Hz for 5 seconds. During a depolarizing blockade, muscle response is sustained. On the other hand, fade occurs during non-depolarizing block and phase 2 blockade of depolarizing muscle relaxation. Post-tetanic count. During profound non-depolarizing muscular blockade, there may be no responses to train of 4 or single twitch stimulation. In such cases, a post-tetanic count may be useful. If a 5-second tetanic stimulus at 50 Hz is administered, after no twitch response has been elicited, followed 3 seconds later by further single twitch at 1 Hz, there may be response to single twitch stimulation. This is known as post-tetanic facilitation. And last one is double burst stimulation. 2 short bursts of tetanus at 50 Hz at a supramaximal current are applied. Each burst will have 3 impulses lasting for 0.2 milliseconds, and each impulse is delivered every 20 milliseconds, and 2 bursts are separated by 750 milliseconds. In muscle partially paralyzed with non-depolarizing muscle relaxation, the response to the second burst is reduced. That is third phenomenon. And double burst stimulation ratio is the ratio of magnitude of second stimulus to the magnitude of first stimulus, which have very similar properties to train of 4 ratio. Train of 4 less than 0.6 or lower can be detected by using this double burst stimulation method. Sir, that's all, sir.
That's all. Okay. What is the question? Can you read the question again?
Sir, physiology of neuromuscular junction and techniques of monitoring neuromuscular junction transmission.
Very good. So the technique is inadequate in your answer. The neuromuscular junction physiology, what you have described, is quite adequate, but the techniques of monitoring, Dr. Ammu, are you there?
Yes, sir.
My contention is that this description of technique is inadequate. Do you agree with that or not?
Just monitoring is mentioned in this, sir.
Okay. Dr. Venkateswar Rao, are you there?
Yes, sir. Yes, sir.
Do you agree with my statement that this description of techniques of monitoring the neuromuscular transmission is inadequate?
Neuromuscular monitoring.
Techniques, is it adequate what he has written? He described in detail about the various methods of stimulation and their
Yes, sir.
But the techniques, does it end with that? Jay Bhardi, are you there? Huh? Anyone who think in my line and say that something more to be written?
Muscular stimulation, MVP, etc. can be seen as
MVP is not for.
It's like neuromuscular monitoring.
That's not actually neuromuscular. But anyway, I'll just show you what I feel is to be added to this answer. So,
Okay.
So, first part is the physiology part, physiology of neuromuscular transmission. Okay, the structure of neuromuscular junction of a skeletal muscle, smooth muscle, cardiac muscle vary a little from each other, but all of them have the more or less the same structure. There is a motor nerve ending, which is termed as the presynaptic part. There is a postsynaptic part, which is made up of motor end plate or part of the muscle membrane. And between these two, there is a cleft, which is called the synaptic cleft. and the area, that is the area between the motor nerve ending and the muscle motor end plate. So you must be able to, though not in this detail, you must be able to draw this line, and the main point that you have to draw the structure in your answer. So you have to make sure that the myelin is getting not there at the terminal portion, and it is covered by Schwann cells on the other side, and it has got. So when you describe that, you must describe about how the vesicles are formed, how the acetylcholine is produced, and how it is released, and how it goes and occupies. All these things have to be described in detail, especially the importance of calcium, which has to enter here and then make the vesicles come to the towards the synaptic cleft, and by exocytosis release all the acetylcholine, and so many millions of acetylcholine are released by every single transmission. And this neuromuscular transmission is mainly an electrochemical transmission. So the electrical part comes up to that where there is a myelin. Beyond that, it is chemical, where acetylcholine is released into the synaptic cleft, and then it goes and occupies the receptors on the motor end plate side. And so this is much more a simpler diagram, which you all can be easily drawn in your exam answer papers. So where you have to give this motor end plate cleft, which is there, the trough and the summit, where the acetylcholine receptors are produced and situated, and at the bottom in the trough you have the acetylcholine esterase enzyme to break the acetylcholine that is released every time. And then the reuse. Sometimes in the exam they ask you about the acetylcholine synthesis and its breakdown. So in the presynaptic neuron, the combination of acetyl-CoA with choline, that forms the acetylcholine, and then it is stored in the vesicles, and once it comes into the cleft, it is broken again by acetylcholine esterase into acetate and choline is reused for further manufacture of acetylcholine. Okay? So this point also you can mention in your answer. And when an impulse from the peripheral and central nervous system reaches the presynaptic membrane, that is the nerve terminal of the neuromuscular junction, in the form of action potential, it triggers. First thing that happens is a voltage gated calcium channels at the active zones of the nerve terminal open, and calcium ions enter into the nerve terminal from the extracellular space. That is the first reaction that happens. The increased intracellular calcium interact with snare proteins. This stimulates synaptic vesicles to fuse with active zones and release their content, that is acetylcholine, into the synaptic cleft. The third step is the released acetylcholine travels across the synaptic cleft towards the motor end plate and binds to the nicotinic acetylcholine receptors, triggering acetylcholine gated channels to open. So this picture tells you about all these things. The earlier picture, this is a much more detailed picture. The myelin stops here, and then the first thing is the calcium entry, first step. Then it makes the acetylcholine get released, and the acetylcholine comes and occupies. Two molecules of acetylcholine have to occupy which component of the nicotinic acetylcholine receptor, which subunit. The two alpha subunits should be occupied, then only the channel will open. So there are 5 subunits in any acetylcholine receptor molecules. So there will be the 2 alpha units have to be occupied by the acetylcholine. Then the channel opens, and then the depolarization occurs, and it spreads to the side to stimulate the entire muscle. The motor end plate on the muscle membrane, the fourth step, becomes more permeable to sodium ions. This changes the membrane potential from -90 to -45 millivolts. This decrease in membrane potential is called the end plate potential. All these terminologies have to be described when you talk about the neuromuscular transmission physiology. In the skeletal neuromuscular junction, the end plate potential is strong enough to propagate action potential over the surface of the membrane. This potential is carried alongside the muscle fiber through the system of three tubules, which triggers release of calcium ions from the sarcoplasmic reticulum into the sarcoplasm, which results in the contraction of the entire muscle. So the remaining acetylcholine in the synaptic cleft gets hydrolyzed by the enzyme acetylcholinesterase. Interestingly, this presynaptic neuromuscular junction, the nerve terminal, also has a nicotinic acetylcholine receptors. So this receptor sends acetylcholine in the synaptic cleft by a feedback mechanism. That is how it is controlled. Otherwise, one single stimulus can be going on releasing all the acetylcholine. So in order to prevent that, there are acetylcholine receptors situated in the nerve terminal also in the presynaptic junction. So the excess acetylcholine that is released goes back and closes that release of acetylcholine from the presynaptic junction. This is an important feedback mechanism. So if the concentration of the acetylcholine in the synaptic cleft is increased appropriately, the presynaptic acetylcholine receptors will sense that and the nerve terminal will shut down more release. This is how each signal is terminated so that it doesn't go on be activating the postsynaptic junction. The difference between pre and postsynaptic acetylcholine receptors is the response of these receptors to different receptor agonists and antagonists. So these receptors, the presynaptic receptors, are not blocked by your muscle relaxant. It is only the post-junctional or the post motor end plate nicotinic receptors which are blocked. So in the exam, if you want to write a very brief and a very fast answer to complete your answer within the time interval, you start from here, motor nerve fiber. There is an action potential stimulated. This results at the axon terminal, opening of voltage gated calcium channel, entry of calcium ions from ECF into the nerve terminal, opening of vesicles to release acetylcholine. This is what is happening at the axon terminal. Then the acetylcholine passes into the synaptic cleft. Then the acetylcholine goes to the postsynaptic membrane, binding with the acetylcholine receptors, and acetylcholine complex forms. This opens the ligand-gated sodium channel. This causes the entry of sodium ions from ECF, development of end plate potential, that is minus 60 millivolts. Then what happens in the muscle fiber? Aggregation of a generation of action potential, excitation, contraction, coupling, and muscle contraction. So 5 steps, 1, 2, 3, 4, 5, what all I have described here, except that there's a feedback mechanism. This is a simpler way of you to write the answer, and you can get equally good marks by saving your time also. Now coming to the monitoring, neuromuscular monitoring is a good guidance whenever there is a need to use neuromuscular blocking agents so that you know whether you have given adequate blockade, and at the same time, especially for intubation and relaxation required for intubation, and you can at the end of the procedure for deciding whether there is any residual blockade left after you have given or whether it is time adequate correct time to give the reversal, and then find out whether the reversal is complete or if there is any residual to the muscle block still present. So that is the idea of giving or monitoring the neuromuscular function. The residual neuromuscular block is associated with serious complications like risk of aspiration pneumonia and hypoxemia, airway obstruction, or need for tracheal reintubation, and patient discomfort, as well as surgeon also looks at you as though you have done a great crime, and increased length of stay in the post-anesthetic care unit. All these things are the complications of inadequate muscle reversal. So antagonism dosage and injection time also can be optimized by proper monitoring of the neuromuscular blockage depth. Now coming to the methods of neuromuscular monitoring, it has to be classified by clinical test, consists of evaluation of the respiratory parameters and muscle function, which are 5 seconds head lift and hand grip. This is the common clinical method by which we assess the re-establishment of neuromuscular monitoring. The second is qualitative evaluation, which employs peripheral nerve stimulator. This is only one part of it and assess the visually or by touch, tactile method, the response of stimulated muscle. This is qualitative evaluation. So for qualitative evaluation only, we use what is called the peripheral nerve stimulator, which provides several patterns of nerve stimulation like train of 4, double burst, tetanic, post-tetanic count, and evaluation of the train of 4 for degree of fade also. Now you can just draw a few diagrams like this and complete your answer for that type of stimulation, single twitch, train of 4. All these things are available in all the books. So you can just draw this so that you need not have to write in detail and waste your time. So for better understanding, I have given another diagram as to how to find out when there is no drug. This is the first train of 4. All the 4 twitches are of equal height, and once the non-depolarizing block is there and you test for fade, you find the first twitch is very tall and the fourth twitch is the shortest. So there is a gradual decline in the height of the muscle contraction and twitch. When it is phase 1 or phase 2, it is a miniature form of the normal that is in phase 1, and but when the depolarizing muscle relaxation goes into phase 2, it behaves like a non-depolarizing and there is a fade occurring there. Okay? So double burst, how the fade is occurring, there is no fade, equal but smaller thing, and again the same response will be there, just like train of 4 what we described. There will be a shorter version of that. So by just drawing these lines, you can save time and mention that fade can be elicited both with train of 4 as well as double burst. And in post-tetanic potentiation, you find there is a decrease, and then the first twitch after the tetanus stimulus is always larger and higher. So that is what is called the post-tetanic potentiation. So by just drawing these lines, which you can save time, you can explain that. And for the newcomers or juniors who have not seen, this is the commonly used peripheral nerve stimulator machine with various switches and the. Can anyone tell me this portion? What is it called? Ammu or Venkateswar Rao? It's black and red.
Black is away from the patient electrode, sir.
No, what do you call this? This part of the equipment. Anyone? Tilak? Mohit? Jagdish? This is called a crocodile clip. See, when you press that, it will open. And when do you use this? This is used when you attach an ECG electrode. So that button you have to just clip it on. So this is called the crocodile clip of the lead, electrical lead. And this is the bulb electrode. Okay, this is for a single time use. You can use the same thing, and you can, if you want to replace that, you have to remove this and then you can attach this, where you have this crocodile clips also available for continuous measurement. So whenever you want to do a single time measurement, instead of attaching the electrode and then connecting this, you can use these two, what is called the bulb electrodes or the ball electrodes, and stimulate the nerve in the same way for a single twitch. Okay. Then the third thing is quantitative monitoring. Uses of neuromuscular monitors, devices that stimulate the peripheral nerve while also recording, quantifying, and displaying numerically the evoked responses. This is the quantitative monitoring. For that, you have all these monitoring techniques called mechanomyograph, electromyograph, and there are totally 6: acceleromyograph, kinomyograph. These are the various methods of or instruments that are used. And lastly, the phonomyograph and compressomyography. So these are all the things which are available. Some of them are used for regular intraoperative monitoring also, some of them more for research purposes. Okay. So the sites where you can use as he mentioned, is commonly we use the ulnar nerve, which results in the adductor pollicis muscle response. That is, thumb adduction will happen. Adductor pollicis response. So the resulting action is the thumb adduction, and where you have to place the electrodes for that is the black 1 to 2 centimeter proximal to the wrist crease, and red 2 to 3. So red should always go towards the body, the black towards the fingers. So remember that way. And now you can stimulate facial nerve also, orbicularis oculi muscle and corrugator superciliae, that is in the forehead region. These are the two muscles which will be stimulated, and twitching of eyelid and eyebrow will happen. And where you have to place the black just anterior to the tragus, and red or white here lateral to the outer canthus of the eye. Then the posterior tibial nerve is the third common nerve where you can just stimulate that. This results in flexor hallucis brevis muscle contraction. So there will be plantar flexion of the great toe. So the great toe will bend down, and the over posterior aspect of medial malleolus you have to place that, and then it should be proximal to the black. So this is how you place the electrodes also. And if you put it them in the opposite, the effect will not be good. Muscle will only get what is called hyperpolarized, and it will not react properly. And common electromyograph which you uses the electrodes which are commonly used for ECG monitoring, and it measures the amplitude of compound muscle action potential, and affected by electrocautery and considered as a gold standard by some experts. So electromyograph is considered as a gold standard for monitoring the neuromuscular junction function. And kinomyograph, it uses what is called the piezoelectric polymer sensor in the groove between thumb and index finger. So this is the portion where the piezoelectric crystal containing sensor is placed, and there is a clip attached to the thumb as well as the index finger. It measures the movement of muscle, which generates voltage in the sensor, and it is less reproducible than the electromyograph. So the electromyograph is the most important thing that is usually done. And in the phonomyography, there's a high fidelity narrow bandwidth microphone placed alongside the muscle. You can see this is the microphone containing sensor which is placed on the thenar eminence muscle, and there is it measures the sound intensity with low clinical use and unclear feature. Then accelerometry is a again uses a piezoelectric crystal and measures acceleration of the muscle which generates voltage in the crystal, and reliably detects the train of ratios, but sometimes can be overestimated compared to EMG. So this is how the accelerometry is placed on the index and the middle finger along with the thumb. And then it measures the voltage generated. So whenever you use piezoelectric, it measures the voltage of generated during the muscle movement. And the mechanomyograph is a force transducer and uses force of contraction, again widely considered as gold standard, but rarely used clinically because elaborate setup and bulk equipment. Because of the bulky nature of the instrument, it is not usually used. But electromyograph is a gold standard which can be used during surgery also. And lastly, there is what is called a compressoromyography. This device incorporates objective monitoring within the blood pressure cuff and has been recently developed and also referred to as modified cuff technique. Cuff-based monitoring appears inspired by compressor myography, but now defunct monitoring modality that showed initial promise. So this is how this compressor myography uses something like a blood pressure cuff itself, and then you can see the display that is happening in the you can see the this is the oxygen saturation and pulse rate. This is the display of the contractions. So these are all the various methods which are qualitative assessment. I mean quantitative assessment. Earlier we said about quantitative assessment, that is the peripheral nerve stimulator. So the three things you have to write whenever you are asked about how to monitor the neuromuscular junction. One is the clinical method, second is the quantitative, and third is the qualitative measure. Okay, so all these things have to be included in your answer.